Okay, students, this is our another lecture of genetics, and today we are going to discuss the chromosomal aberrations, chromosomal mutations, and in chromosomal aberrations or chromosomal mutations, we gonna discuss first example, that is Down syndrome. So my topic is Down syndrome, right? So let's start. So what is Down syndrome? This Down syndrome is also known as 21 trisomy. It is also known as 21 trisomy. So please try to understand 21 trisomy means where the chromosome number 21 is 3 times. So instead of 2 times, the chromosome number 21 exists 3 times. It occurs 3 times. There are 3 copies of chromosome number 21, right? That is why it is known as 21 trisomy. I will explain everything. Anyways, this Down syndrome, it is also known as Mongolism. It is also known as Mongolism. This is one of the symptom of the disease where the individual with this Down syndrome is having epicanthal fold. That means the upper eyelids are turning downwards, and apparently this individual is like a Mongolian individual, but they have a particular race. Mongolians have a particular race, that is their racial character. This is the problem with the individual, but apparently this individual with the Down syndrome is like a Mongolian individual, right? So that is why this Down syndrome, it is also known as Mongolism, or it is known as Mongolian idiocy. Because this individual is not normal individual. It's not normal individual. It's not having these problem with the eyelids, right? It is also mentally retarded. This individual is also mentally retarded. There is mental retardation in the individual, so that is why idiocy. So this is known as Down syndrome. This is known as 21 trisomy. It is also known as Mongolism. It is also known as Mongolian idiocy, right? Now let's try to understand what is this disorder. This syndrome, it is actually the genetic disorder. If we like to describe this, it is a genetic disorder. It's a genetic disorder in which the chromosome number 21 is present 3 times. The chromosome number 21 occurs 3 times instead of 2 times. There are 3 copies of 21 in the genetic disorder instead of 2 copies. So we can say there are 3 copies of chromosome number 21. So there are presence of 3 copies of chromosome number 21 in the genetic disorder. How they occur, how they exist, we will explain this ahead. So this is the precise definition of the Down syndrome. This is a genetic disorder. It is a chromosomal abnormality, we can say also, in which the chromosome number 21 exists, or it is present 3 times, or there are 3 copies of this chromosome number 21, right? Now, so far as these chromosomal mutations are concerned, this Down syndrome or 21 trisomy, it is one of the most common chromosomal abnormality. One of the most common chromosomal abnormality in humans. So among the chromosomal abnormalities where 1 or 2 chromosomes are extra or they are deficient, right? This 21 trisomy is one of the most common chromosomal abnormalities. Anyways, now it was first discovered, this Down syndrome 21 trisomy was first discovered by a British doctor. It was discovered by a British doctor whose name was John Langdon Down. It was first discovered by this British doctor, physician, whose name was John Langdon Down, on the basis of symptoms. He was not aware about the cause, what is actually the molecular cause of the genetic cause of the Down syndrome. He was not aware about that, but on the basis of symptoms, different symptoms, he called this disease Down syndrome. He used his own name. So that's why this disease is, this disorder, this syndrome is known as Down syndrome. It is actually this British doctor who discovered this syndrome. Anyways, now so far as the cause of this disease is concerned, why there are 3 copies of 21 when all the chromosomes, other chromosomes, the rest of the chromosomes, they occur two times. I mean, there are 2 copies of each chromosome, but only the 21 is 3 times. So what is the cause for this 21 trisomy? So let us try to discuss the cause of Down syndrome. So as I told you that Langdon Down, who discovered the disease on the basis of symptoms, was not aware about the cause. What are the reasons for this 21 trisomy? So it was not described by the Down what is the real cause of the 21 trisomies. So it was another doctor whose name was Lejeune in 1959, most probably. Lejeune was another biologist who actually described the cause of Down's syndrome. And it is this Lejeune who found that in this individual with the symptoms of Down syndrome is having 3 copies of, he found that there are 3 copies of chromosome number 21. Let me tell you again. The cause was described by the Lejeune, and he found, after developing the karyotype of the individual, counting the number of the chromosomes of the individual in his or her cells, he finally confirmed that there are 3 copies of chromosome number 21 instead of 2. So the total number of the chromosomes in this individual with Down syndrome is 47. So one chromosome is extra, one copy of the chromosome is extra. It should be 46. You know that the normal chromosome complement of the humans, whether it is male or female, but it is 47. In this condition, the chromosome number is 47. But again, the question is, which chromosome is in extra? So the chromosome number which is in extra, whose copy is extra, is again this chromosome number 21. So there are 3 copies of 21 instead of 2. You can understand better this point if I will show you the other chromosomes as well. For example, we have developed the karyotype. Karyotype means where we see, where we identify, where we determine the shape, size and the number of the chromosomes, right? For this we can go for the different diagnostic or analysis, right, where we isolate the chromosomes of the cell, and then we can get the photograph of the chromosomes, and then we can count, and even we can find the shape, size of the chromosomes. This particular aspect of the genetics, human genetics, is known as karyotype. If, for example, if we show the karyotype of this individual who is with Down syndrome, so we can say, for example, this is chromosome number 1. Let's say it is chromosome number 1. Chromosome number 1. There are 2 copies of chromosome number 1. And let's say it is chromosome number 2. So 2 copies of chromosome number 2. This is chromosome number 3. This is chromosome number 4. This is chromosome number 5. This is chromosome number 6. This is chromosome number 7. Okay. This is chromosome number 8. If you can see here, we can show this is chromosome number 9. This is chromosome number 10. This is chromosome number 11. This is chromosome number 12. This is chromosome number 13. This one is chromosome number 14, 15, 16, 17. Okay. This is chromosome number 18. This is 19, 20. This is 21. Please try to understand. This is chromosome number 21. So we have 3 copies of 3 homologs of 21 in a state of 2. So here is the problem. Here is the problem. That is 21 is present 3 times in a state of 2. You can see all the other chromosomes, whether it is chromosome number 1 or it is, for example, chromosome number 9 or, for example, 18. They all exist in 2 copies. But chromosome number 21 in this individual is present 3 times. If we write other chromosomes, for example, 22, for example, that exists in 2 copies, 22. Then we have the X or XY, for let's say it is male. So X and Y are also present 2 times, 2 copies. One is X, another is Y. But here again, please try to focus at this point, that 21 chromosome which should be present 2 times, it is present 3 times. There are 3 copies of this 21. So this is actual cause. This is the actual cause of this Down syndrome. There are 3 copies of 21. That is why this is known as 21 trisomy. So if you will develop the karyotype of the individual, you will find there are 3 copies of chromosome number 21. So that means 1 chromosome of the 21 that is extra, that is having the extra genes in this individual. There are extra genes of that chromosome in the individual, which is actually responsible for this disorder and showing the different types of symptoms in Down syndromic patient, right? Now here again the question is why this 21? Why this 21 is 3 times? Why in this individual with Down syndrome there are 3 copies of 21 when all the other chromosomes are present only 2 times? I mean, there are 2 copies of each other chromosome, right? Now the answer to this question is this condition where the 21 chromosome is 3 times, there are 3 copies of 21, is because of occasional. It is because of occasional non-disjunction. Occasional non-disjunction. Why an individual is having 3 copies of chromosome number 21 in a state of 2. The reason is occasional non-disjunction. We have to understand this point. What do we mean by non-disjunction? And in order to understand this point, non-disjunction, we have to focus on meiosis at the time of gametogenesis. Meiosis that occurs at the time of gametogenesis. So generally what we see, for example, this is a germ cell, for example, in male or a female, and having 46 chromosomes, right? Each chromosome is present 2 times. There are 2 homologs of each chromosome, even XX or XY, right? Now, and 21, if I, for example, show here the 21 chromosome, this 21 chromosome is also present 2 times. There are 2 copies of 21. Let's say this chromosome is 21. What normally happens? What normally happens? Let's say this cell is undergoing the meiosis, and first there will be meiosis first. There will be meiosis first, and then there will be meiosis second. Now, normally what happens? This cell will divide into two cells. Please try to understand. This cell will divide into two cells. This will be one daughter cell. This will be another daughter cell. And normally what happens? Disjunction. Disjunction means this chromosome will be received by this cell and this chromosome will be received by this cell. That means these two chromosomes of 21 will be equally distributed in each of these daughter cells. One will go to this cell, another will go to this cell. This is normal division. So if I will show this division normally, so this chromosome, let's say this one, this chromosome will move to this cell, okay, and this chromosome will, another chromosome will move to this cell. So these two cells, these two daughter cells after the meiosis first have received one copy of 21. Likewise, they have received the one copy of each other chromosome, chromosome number 1, 2, 3, 4, so forth and so on. So I can say this cell is having 23 chromosomes and this cell is also having 23 chromosomes. There is one copy of each chromosome and one copy of 21 here, one copy of 21 here. Now if they will divide further, for example, this cell will again divide in meiosis second, this will divide in meiosis second and it will give rise to cells again. Again there is no problem because this is the normal division, and here the chromatids will separate from each other. So this will receive one chromatid, this cell will receive one chromatid, right? Okay. Again here this cell will divide into two daughter cells, and again there is no change in the chromosome number. This will receive one chromatid, one chromosome, this will receive the another chromatid. So these are the four haploid cells, N, N, N and N. These are the four haploid cells. They have received, all of these daughter cells have received 23 chromosomes. That means in each four daughter cells there is one copy of each chromosome, whether it is chromosome number 1, 2 or for example 21, right? So this is normal division. We can say it is disjunction. There is separation of the chromosome number 21 and they get separated from each other, and each daughter cell in the meiosis first receives one copy, one homologue of the 21. Then there is further division of these two daughter cells which are formed in the meiosis first, and then again there is equal separation of these chromatids and each four daughter cells is receiving the one copy of 21. This is the normal phenomenon of disjunction. This is normal phenomenon of meiosis. But what happens in non-disjunction? Our point is non-disjunction. My point is non-disjunction. What happens in the non-disjunction? Let's try to understand. In non-disjunction, let me rub this first. In non-disjunction, what is the problem? Let's say again this is the same cell which is undergoing meiosis first and second. Again, let's say this is chromosome number 21 here. This is the chromosome number 21 here and it's two copies of 21. Likewise, other chromosomes are 2 times here in this cell. So total number of chromosomes in this cell are 46. Now see, generally what happens in the meiosis first? Generally meiosis first, but it can, this non-disjunction can happen in the meiosis second as well. But generally it happens in the meiosis first. So what happens here? See, this is one daughter cell, this is another daughter cell. Meiosis first took place, meiosis first completed, right? Now normally what should happen? Yes, normally what should happen? This chromosome should go to this cell, this chromosome should move to this cell, but that is not happening. Then what? These both chromosomes, these both copies of 21 are received by the one cell, this cell for example. This cell is receiving both the copies of 21. This cell has received one copy of all other chromosomes, chromosome number 1, 2, 3, so forth and so on. But there is problem with the disjunction of 21. Both these 21 chromosomes, both the copies of 21 will go to the one cell. So this cell is receiving one extra copy of chromosome, and that one extra copy is 21. So if you see in this daughter cell, there are 24 chromosomes, one is extra. There are 24 chromosomes, and this cell, it has lost its one chromosome, that's 21 copy. It's not having any copy of the 21, so there will be only 22 chromosomes in this cell, 22 chromosomes in this cell, but there is no 21 in this cell, in this daughter cell. Okay, because these two chromosomes which should have that should have separate, and each should go to the each daughter cell, but both the chromosomes are moving towards the one cell. So this cell has received both the copies of 21, so the total number of the chromosomes in this daughter cell are 24, while this cell is containing only 22. So if it will further divide, for example, it will go to the meiosis second, the problem will remain there again. We can get two daughter cells again from this cell. There will be no change in the chromosome number, because meiosis second is almost equal, similar to mitosis. So these both copies will remain as such. So here we have the two copies of 21 again. Here we have again the two copies of 21. So again this cell, it is having 24 chromosomes, this cell is having 24 chromosomes, and the extra copy is 21 in these two daughter cells. While this cell, again there will be no change in the chromosome number, right? So this cell, again there will be 22 chromosomes. Here again there will be 22 chromosomes, but you know this, these two daughter cells which are developed from this cell, which has already lost its 21 in the meiosis first, right? So again these two daughter cells formed from this cell do not have the 21, they have lost their 21, right? But these cells which developed from this cell, which has received the 21 already in the meiosis first, there are two copies of 21. So again these two cells have the 24 chromosomes, and the chromosome copy which is in extra is 21. Now, for example, these two cells which are having an extra copy of 21, please try to understand. These two cells, I am talking about these two cells. If these two cells, for example, develop into sperms, let's say they develop into two sperms. This sperm is having 24 chromosomes, this sperm is having 24 chromosomes, and again we know this. These two sperms which developed from these two cells where the non-disjunction took place, this chromosome is having one copy of each chromosome number 1 to 23, but one copy is extra in this sperm and in this sperm, that is 21. So there are two copies in this sperm, there are two copies in this sperm, two copies of what? 21. Two copies of 21. So these two sperms have one chromosome in extra, and that one chromosome which is in extra is 21. Instead of one copy, they have two copies of 21. Let's say this sperm, among these two sperms, one is fertilizing a normal egg. Let's say this is a normal egg which don't have any non-disjunction, which don't have any chromosomal abnormality. So this egg, it is having 23 chromosomes, and obviously if it is having 23 chromosomes, there is one copy of 21 in the egg. Try to understand. These sperms developed from these cells where the non-disjunction took place in the meiosis first, in chromosome number 21. So as a result, these two sperms are containing two copies of 21. If now, for example, this sperm is used in the fertilization, this will fertilize a normal egg. It is fertilizing a normal egg. So there are two copies of 21. Here we have one copy of 21 in egg. So what will be the chromosome complement of the zygote? Yes. What will be the chromosome complement of the zygote? The chromosome complement of the zygote will be 47. Total number of chromosomes in the zygote will be 47, and the zygote will receive two copies of 21 from the sperm, 1, 2, and it will receive one copy of chromosome number 21 from the egg. So there are three copies of chromosome number 21. That is the cause of the genetic disorder. This results the genetic disorder what we call as Down's syndrome. As a result of these 3 copies of 21, there will be different types of symptoms and signs in the Down syndromic patient. Please try to understand how this happened. Actually, the problem occurs here in the meiosis. When the meiosis first took place, right, this chromosome number 21, which is existing as 2 copies, it should separate from each other. They should separate from each other, and each homologue should be received by the daughter cell. But what happened? Both these copies of 21 are received by the one cell, while the other cell is lacking the 21. Now we are talking about this cell which has received an extra copy of 21. It will further divide into 2 cells, and finally 4 daughter cells are formed, but these 4 daughter cells are different. These 2 cells, first 2 cells, they have an extra chromosome, that is 21, 2 copies of chromosome number 21. While these 2 cells are lacking the 21, they don't have any copy of 21. Now we are supposing that, for example, if this cell will develop into a sperm, it can develop into an egg. That is also the case, right? So for example, let's say this cell will divide into the sperm. Now this sperm is having an extra copy of 21. In sperms are haploid. They should have 1 copy of each chromosome, but they have 2 copies of 21. And let's say this chromosome will fertilize a normal egg. Egg is not having any problem. So if it is fertilizing a normal egg is having 1 copy of 21, the net result will be 3 copies of 21 in the zygote. So there is an extra copy of chromosome in the zygote, and that extra copy is 21. 21 should be 2 times in the zygote, but that is 3 times. So that is Down's syndrome. Clear? So this is actual cause of this Down's syndrome. But let me add here, because I'm not gonna discuss this again and again in other chromosomal mutations, in other chromosomal abnormalities. There are many other chromosomal abnormalities that we have to discuss in the upcoming lectures. For example, we have Klinefelter syndrome, we have Turner syndrome, we have many other syndromes as well, but these 2 we have to discuss. We are supposed to discuss these 2, Klinefelter syndrome and Turner syndrome. Again, these are chromosomal aberrations, these are chromosomal abnormalities, and in both these genetic disorders that we will discuss in future, there is again the problem of non-disjunction. If there is any chromosome in extra, that is extra copy of chromosome 1 or 2, or any chromosome is lacking in the chromosome complement, that is again because of the same cause, that is non-disjunction. So you have to understand this point here, that these chromosomal abnormalities like Down's syndrome, 21 trisomy, they all results due to the non-disjunction, where the chromosomes, homologous chromosomes, fail to separate from each other, and both the chromosomes move to the one cell while other cell is lacking that chromosome, right? So this is the actual cause of the Down's syndrome, right? Now if we talk about the different signs and symptoms in the Down's syndrome, what will be the different signs and symptoms? There are so many symptoms, there are so many complications in the individual with Down's syndrome. Let's discuss some of the important signs with the help of which you can diagnose, you can recognize a patient with Down's syndrome. So let's write here signs and symptoms. How you can locate a person from the distance without doing the test, without doing the diagnosis that this individual is with Down's syndrome. The most important sign that you can observe from the outside body, right, is epicanthal fold. Epicanthal fold. The sign means, epicanthal fold means, that is the upper eyelids. In this patient, upper eyelids turn downwards, right? almost closed type of eye, closed eyes. There is a little space between the two eyelids, almost like an individual, the individual from the Mongolian race. That is why this is also known as Mongolism or Mongolian idiocy. But this is a defect, you know this. So this is first sign, that's known as epicanthal fold, right? Okay. Another sign is that this individual with Down syndrome, it is generally most of the babies with Down syndrome have a happy look. Happy look. They look happy. They are not really, but this is their mental problem. This is their problem with the brain, right? So they have happy look. Number 3, they are mentally retarded. Yes, they are mentally retarded. They don't have the IQ of the normal individuals. They have low IQ. They have low intelligence coefficient. So they are mentally retarded. They are mentally retarded. They are mentally retarded. Okay, fine. They are mentally retarded. Fourth sign that you can see is they have broad hands. They have broad hands. Okay, they have broad hands. Hands are bigger in size. And number 2, that their palm, there are some crease in the palm. Generally they are 2 or 3 in number, right? But in these individuals there is only single. This is the 5th sign. There is single crease of palm. Single crease of palm. Right? If you look at the palm of the individual, you will see only one crease there. So single crease of the palm. Number 2. So far we have discussed how many signs? 5 signs. Let's move to the 6th one. Let's write here. 6th one is that these individuals have underdeveloped genitalia. They have underdeveloped genitalia. Okay, they have underdeveloped genitalia, whether male or female. Now, one more sign is that these individuals have some congenital from birth, congenital defects of heart defects of heart defects. Generally, there is problem with the septums. Ventral septums are defective. Ventricular septums are defective in these individuals. Ventricular septums, right? So this is one more thing. And next thing is that these individuals are prone to the cancers like leukemia. They are prone to the blood cancers. Blood cancers can occur in their body during their lifetime. And overall, these individuals have short life expectancy. They have short lifespan than the normal individuals. And one more thing that I would like to add here is the gene. There is a gene on the chromosome number 21. That gene codes for a protein. That protein is known as amyloid protein. This amyloid protein, it increases and it accumulates in the aged people generally, and then they suffer from a disease that's known as Alzheimer's disease. Again, a mental problem, Alzheimer's disease. But these individuals from their beginning, from their early life, they have the one copy of 21 in extra, and that 21 chromosome number is carrying a gene for amyloid protein. So that is why what we see in most of these individuals with Down syndrome, they also suffer from this Alzheimer's disease. Alzheimer's disease. There is Alzheimer's disease in these individuals due to the accumulation of this amyloid protein in their brain cells. because the gene of this amyloid protein is also present on the chromosome number 21, which is extra in the individuals of Down syndrome, right? So this is one more sign. I told you that there are so many complications, there are many signs and symptoms in the individual with Down syndrome. These are the most important ones, with the help of which you can locate a person, you can recognize a person with Down syndrome. Hope you have understood. Now, so far as screening is concerned, I mean diagnosis, can we diagnose this with the help of any diagnostic technique? For such individuals, we can go for prenatal. For such individuals, we can use prenatal screening. We can use prenatal screening. That is done in many countries. Once there is birth of the baby, and they go for the prenatal screening, and they confirm whether their baby is not having this Down syndrome, right? So prenatal screening, in prenatal screening we can use some techniques. For example, again, chorionic villi sampling, CVS, chorionic villi sampling, where some chorionic cells are extracted from the individual, from the embryo, right? And then the chromosomes of the cells are isolated, and then karyotype. After karyotype, we can see whether the number of chromosomes is correct, or if there is any extra chromosome like 21. We can go for also amniocentesis. Amniotic cells can be used to find the exact number of chromosomes. Amniocentesis, or we can say amniotic fluid test. So this is one more thing, right? Genetic screening is also there. With the help of these different screening techniques, diagnostic techniques, we can determine whether this baby which is going to born, right, is having this Down syndrome or not, right? So this is prenatal screening. Now, so far as treatment, can we provide any treatment, any cure to the patient with Down syndrome? No, there is no cure. Since this is chromosomal abnormality, so it is very much there. There is no cure. No treatment for this individual. Yes, they can have different Down syndromic patients can have different lifespans. Some have shorter, some have good lifespan. I mean, they can live for many years, adult life, right? Depending upon the complications. Okay. Now, then what we can do for these individuals? We can help these individuals. We can manage their condition. So we can give them, we can provide the proper care. By providing the proper care to these individuals, we have not to ignore them, right? So if we will not ignore them, if we will not let them to go astray, right? So we can take proper care of them, and we can help, in this way we can help these individuals with Down syndrome. Okay. Now, lastly, I would like to mention that sometimes this Down syndrome is familial Down syndrome. That's a different type of condition. So that is why I am discussing this, at last. So there is one more condition that is associated with this Down syndrome, and that is, let me come back. So there is familial Down syndrome. Familial Down syndrome. What does this term mean? Familial Down syndrome. Familial means it run in the family. It can run for 2-3 generations. So that is why familial Down syndrome, because the symptoms will be similar to the Down syndrome, which is because of the 21 trisomy. Now, what is the cause of this familial Down syndrome? The cause of the familial Down syndrome is what? Let's say, for example, this is chromosome number 21. Okay, this is chromosome number 21 of an individual. And let's say, for example, this is chromosome number 14. So this is chromosome number 21, 21 and this is chromosome number 14. Okay, 14. Sometimes what happens, it is having a long arm, this is say Q and this is P. P is short arm, please keep in mind. When we use two arms in metacentric chromosomes, where the centromere lies at submetacentric chromosomes, where the centromere is not exactly at the center and the two arms are not equal. So we say one arm is longer and another is shorter, short arm is P and longer is Q. And what happens sometimes in this familial Down syndrome, this long arm of the what 21 is translocated to this translocated. It is translocated. There is translocation of long arm of the Q arm of the what chromosome number 21 to the 14. And when this happens, let's say for example, there is loss of the this is the P arm of the chromosome number 21. It has lost its Q arm, which has translocated to the chromosome number 14. So we can make this chromosome number 14 a bit longer now, because it is having also 21 Q on it. It has received an arm from the chromosome number 21, right? And what happened to this chromosome number 21? It has lost its arm. There is deletion in the chromosome number 21. Its long arm, that's Q. Sometimes what happens with this chromosome number 21, it duplicates this P arm again. It duplicates its P arm, and there are two P arms. There are two P arms in the 21. Very interesting thing if you can understand. This 21, which has lost its Q arm, long arm, that is transferred to that's translocated to the 14, right? So now the chromosome number 14 is having one more segment of the chromosome number 21 on it, you can see, right? But what happens to 21? 21, it duplicates its P arm, and now there are two P arms. There are two, that means similar arms on the 21. When a chromosome is having mirror image arms, when a chromosome is having mirror image arms on it, that means it should be, one should be P, another should be Q, but both are PP or both are QQ. This chromosome is known as isochromosome. This chromosome is known as isochromosome. Isochromosome is not a normal condition, guys. Isochromosome is an abnormality with a chromosome. When a chromosome loses its segment, loses its arm, then there are chances with that chromosome which has lost its part that can duplicate its existing arm, and that will acquire two similar type of arms, mirror image arms. And when there are two mirror image arms present on a chromosome, this is known as isochromosome. So this will happen with the 21, but you know that what happened to the 14. Now this 14 chromosome is having its own arms, and it is having an additional segment of chromosome number 21, long arm of the chromosome number 21. If you develop the karyotype of this individual with familial Down syndrome, there will be no change in the chromosome number. Chromosome number will be similar. Only the problem lies with chromosome number 14 now, which is having an extra segment of 21 on it, right? So the chromosome number will be 46, but there is translocation of long arm of 21 to 14. So type of partial trisomy is there, but it's not the complete trisomy. You cannot see in the karyotype 47 chromosomes. You will see only 46 chromosomes, but you will see problem with the, when you will see the shape and the size of the 14, you will see it is a bit longer because it has received one long arm of the chromosome number 21, translocation of long arm of the chromosome number 21 to 14, and this is so called as familial Down syndrome. So with this, guys, we have completed this topic as well, that is Down syndrome or 21 trisomy. If you have any problems, queries and clarifications, you can contact, right, and we will solve your queries and clarifications. Thank you for watching.